Hello, I'm Rama Ranganathan, a professor in the Green Center for Systems Biology and also affiliated with the departments of biophysics and pharmacology at UT Southwestern Medical Center. In these three lectures, we're discussing a problem that we call the evolutionary design of proteins. And in the first lecture, I set up the problem by describing the nature of the problem itself. What do we mean by the design of proteins? What is the nature of this problem? And why is this problem interesting and why it's hard? In this lecture, I would like to discuss a proposal of an approach to this problem, a new approach, and then discuss a general model for proteins that emerges from the application of this approach. Now, to review again for what we said in the last lecture, the problem of proteins is the following. Proteins are synthesized as these long polymers of basic building blocks that we call the amino acids. And classical studies show us that the information in the sequence is such that the protein, in physiological conditions anyway, can spontaneously fold up into this precise and ordered three-dimensional structure. That's the so-called folding problem in proteins. But also, information in the sequence directs the ability of the folded state to carry out various biochemical activities. So in the case of this lower protein, the ability to bind substrate ligands at the binding pocket on the surface of the protein. So the fundamental question is the nature of this information encoding. What information in the sequence, constraints on the amino acids and interactions between amino acids, is necessary and sufficient to specify these properties of natural proteins, the capacity to fold, the ability to carry out biochemical activities, and then, of course, since these are natural proteins that are subject to the evolutionary process, the ability of proteins to be adaptive and to change their function in response to changing selection pressures, that is, say, the capacity for adaptation. So what design of proteins accounts for all these properties? And again, I want to remind that by design we mean the pattern of physical interactions between all the amino acids that mediates all of these properties. And in the last lecture, we came to the conclusion that the sort of essence of the problem is that we don't see energy in structures. That is, we don't see the significance or the value of the interactions between atoms that we observe in protein structures, because those interactions are very subtle and the net stability of proteins is quite marginal, and the forces acting between atoms are very detail dependent. That is to say, they're very steeply dependent on distance and geometry, and it's difficult to know the net value of all the interactions, even from high resolution protein structures. So the question is, what approach can we take then to address this problem, the pattern of interactions between all the amino acids that make up a protein? So an approach that we've taken to this problem is a different one from the standard approach. A standard approach would be to start with a specific protein, a single protein, and through high resolution structure determination and experimental measurements on that protein, try to deduce the pattern of these forces acting between atoms. In this approach, which is distinct, we take a statistical approach to this problem. The idea in this approach is to generalize a principle that we all use anyway in our studying of proteins, and that is the principle of so-called conservation. So let me describe this approach to you. The idea is, let's say you want to know which amino acids and interactions of amino acids are important for protein B. In this approach, you don't look at one example of protein B alone. Instead, you go and collect yourself a large and diverse ensemble of sequences that comprise protein B throughout the divergence and evolution. So this represents a protein family, that is to say, an ensemble of sequences found in all different organisms that share the property of folding up and functioning approximately like protein B. And in the limit that you have a sufficiently large and diverse sampling of these sequences, you can make some simple statistical conjectures. For example, if I told you that a position like amino acid K, that position was totally unimportant for anything, for protein B. It doesn't contribute to its folding, its function, or any aspect of fitness. The question is, what should we observe at that position for the frequency distribution of all the amino acids? And the idea, of course, is that such a position should just relax to the mean frequencies of amino acids found in all proteins in nature. The idea is such a distribution is a target distribution, given random mutagenesis, for a position with no evolutionary constraint. On the other hand, if there are positions like I and J that are important for something, then maybe we would observe distributions of amino acids that look like this, say 50% phenylalanine, and intuitively we would say that's a moderately conserved position. And what we mean by moderate conservation in a mathematical sense is that the probability of seeing 50% phenylalanines at that position, given that the mean expectation is like 5%, is very low. That is, the improbability of this outcome at this position is in some sense a measure of the evolutionary constraint for that amino acid at that position. One can simply mathematically write down exactly what I just said in English, which is to say, write down a mathematical parameter that measures the conservation of an amino acid A at a position I as just what I said, the probability of seeing that frequency at that site given the background expectation Q. And the limit of sufficiently large number of sequences, this calculation reduces to a thing that looks like this, which is quite a famous equation in the area of information theory known as the Kullback-Leibler relative entropy, or sometimes called the divergence entropy. What it just means is that this quantity measures the degree to which the observed distribution at the site deviates from the random expectation Q, and is say a measure of the evolutionary constraint at a position. But this is just an analysis of what I would say is a first order statistics. That is to say, the constraint on positions taken independent of each other. But we are looking, of course, to understand the pattern of interactions between amino acids, the way that all the amino acids are talking to each other or interacting with each other through the physical forces acting between all the atoms. And so the question is, how should we think about, mathematically, conserved interactions between amino acids? So let's say positions I and J are interacting with each other, talking to each other in this sense, then what should we expect for the statistical outcome of amino acids at positions I and J? And of course, the idea is quite simple. We shouldn't expect I and J to evolve independently. Instead, we should expect the outcome of J to some extent to control the outcome at I. And in principle, what I'm talking about then are correlations or second order terms in the property of conservation. So let me explain this. So if in this case, what we mean is we should really think about conservation like a hierarchical expansion over terms. So the observed conservation at a position is perhaps some intrinsic pressure on that site to be a particular amino acid. And by intrinsic, what I mean is the pressure on that site to be a particular amino acid taken independently of the outcome anywhere else in the protein. But then, of course, if there are these constraints coming from other positions, we account for them in these pairwise terms, the degree to which an amino acid at one position controls the outcome of an amino acid at a different position. And as I say, these are can be thought of as sort of a second order expansion of conservation to consider the correlated evolution or coevolution of pairs of positions. Now, if you will allow me that, then one can of course ask the question of why not consider then the third order terms, the degree to which three amino acids at different positions might co-evolve in a way which is not just explained by the underlying pairwise coevolutions. Now, if you think about it that way, then you realize that there are quite a number of higher and higher order terms in this view of conservation. But I can say two things. One is that the genome databases, the size they are right now, the number of samples we have of sequences are such that we won't be able to constrain these high order statistics anyway. And experiments are beginning to show us that these second order terms alone actually explain quite a lot what we want to know about protein structure and function, and I'll get to that in lecture 3. So what do these pairwise terms in evolution, the way in which pairs of positions in a protein co-evolve with each other, teach us then about the problem of protein structure, function, and adaptation? So here is that calculation for a particular protein family. So this is an object that we call the statistical coupling matrix, and it's computed for roughly 1,000 sequences, an ensemble of 1,000 sequences of this protein family, the PDZ domain. And let me just introduce the PDZ domain, since it'll be sort of a workhorse system for many of the examples I will illustrate in these lectures. So the PDZ domains are roughly 100 amino acid long protein interaction modules. They're found in many proteins in cells and are often involved in subcellular localization and scaffolding. The PDZ domain adopts a structure approximately of a 6-stranded beta sandwich, as we would say, with a couple of asymmetric helices. And ligands for the PDZ domain, which are typically C-terminal peptides from various target proteins, bind in this groove between this beta strand and this alpha helix and make a number of interactions, which we will discuss later on. What this matrix shows us then is the degree to which every position in the PDZ domain going from N terminus to C terminus is correlated as evolution or co-evolving with every other position going from N terminus to C terminus. So this is a diagonally symmetric matrix where the diagonal terms tell you something about the intrinsic conservation of each of the positions, and the off-diagonal terms, such as this, tell you how much this position of the protein is correlated in its evolution with that other position of the protein. Globally, this is then a view of the pattern of amino acid interactions seen through the lens of evolution, how all the amino acids are co-evolving with each other. Now, we want to understand this pattern. What does this pattern and this matrix tell us about what are the groups of amino acids that are correlated with each other? And to do that, let's begin simpler by just considering one position and the correlations of that one position, just to begin to gain some intuition about the nature of what's information that's in this matrix. So if we pick one position just to start, what that picking one position means is that choosing one column of this matrix, so that's the correlations for that position, which is position 372 in this particular PDZ domain, and defines this amino acid, which is the main function, main determinant of function in the PDZ domain. This is an amino acid position which controls primary specificity for target ligands. The interesting thing is, if you look at the pattern of correlations for this position, you see that they're distributed up and down the chain all over the place. So in fact, their positions are quite far away in the primary structure that are correlated to this position. And the question is, where are they in the three-dimensional structure if we consider the folded protein? And the interesting result was that even if you look at the three-dimensional structure, all of these positions that are correlated in evolution with this position are not just describing the local environment of this position here. So for example, positions correlated to this site are located, some of them are in fact located in the immediate vicinity, which makes sense, but there are other amino acids in blue you see next door to it, which are essentially evolving as if they're independent agents from this position, even though they're quite physically proximal to this amino acid position. And yet there are positions that are at the other side of the binding pocket, and even a couple of positions which are fully on the back side of the protein that look like they are correlated in evolution with this position. So the pattern of coevolution between positions doesn't look just like the spatial proximity around the site, describes instead some pattern that seems potentially more interesting. This actually begs then some kind of approach to globally decompose this information to figure out what is the pattern globally of coevolution between amino acids. How do we do that? Now, to do this, we've actually borrowed methods from different fields. It turns out that the similar problem is experienced by people in statistical physics and actually in economics. And I'm going to describe the solution to this problem through a problem in economics because it's potentially more intuitive and easy to understand. Let me first describe the nature of the analytical problem in extracting information from this matrix. The first goal, the most basic goal, is to separate the functional correlations, the ones that you're really interested in, away from all the statistical noise that contributes to the correlations in this matrix. And let me explain where that comes from. After all, we only have a finite sampling of sequences. We maybe have 1,000 examples of a protein family. And what that means, whenever you have finite sampling of sequences, is that you expect some accidental or spurious correlations to just result from the finite sampling of members in your ensemble. So in fact, I can say that when the number of positions in a protein is not large, or the number of samples of sequences is not large compared to the number of positions, you expect the majority of correlations in these kind of matrix to be dominated by just that noise, statistical noise. So we need to separate the functional correlations from the statistical noise, and then the remaining significant correlations can be examined for some functional patterns of coevolution between amino acids. As I said, this problem has been faced in the field of econophysics. So this is a field of economics, which is a mathematical one. And the goal of this, one goal of this field is to develop, for example, a rational investment strategy for the stock market for optimizing return. And you'll see in a moment why this is a problem that bears a lot of similarity to the analytical problem we face with proteins. So the idea in this approach is to understand the natural breakdown of the economy by looking at how stocks are correlated in their market performance. Now, we all know that the general principle of investment is to diversify our stock portfolio, put on money in as many independent modes of the market as possible to mitigate risk. But how do we identify what are the independent modes of the stock market? Well, one approach might be to deeply know something about all the companies and the way that they interact and so on. But it turns out that one can also pose this problem mathematically by making correlations matrices over the performance of all the stocks in the market. So I show an example here. Here, for example, is the stock price of 3 different stocks. And those of you who invest will recognize that this is Chevron, this is ExxonMobil, this is Citicorp. And what this graph shows you is the stock performance or sampled roughly once a day over a 7-year period. And what you can see intuitively is that Chevron and ExxonMobil seem to be rather similar or correlated in their performance, whereas Citicorp is doing something different. And in general, what this makes sense to us, both these are energy companies, they experience similar market forces. This one may experience different forces and is distinct from the other two. So what people working in this field have done is to make essentially a covariance matrix over the performance of a bunch of stocks over some defined time window here that's 7 years from the S&P 500. And now the important thing to realize is the analogy to our problem. This matrix is also going to be contaminated with sampling noise, because after all, you have a finite number of samples of the stock performance over 7 years, not infinite sampling. So you expect a large number of the correlations between stocks to, again, potentially come from just the finite size of the time series, similar to our problem of finite sampling of sequences. And the approach these investigators took is to carry out a mathematical analysis, which I will not describe in detail, called the eigenvalue decomposition of this covariance matrix. But let me just explain it intuitively. What this graph shows you is a histogram of eigenvalues of the stock market correlation matrix. And the point is that there is a small few eigenvalues in the tail that are quite large, and the bulk of eigenvalues that are all in this region, which are small in magnitude. And the important thing is to understand what an eigenvalue represents. Each eigenvalue is not one stock in the market, but a group of stocks that experience a correlated effect in their market performance. And so each eigenvalue represents a collection of stocks that move together in the market. And the interesting thing one can do is to find the significant eigenvalues, the ones that are relevant, separated from the ones that are just possible through the sampling noise in the data set, by making essentially many trials of a random correlation matrix. What a random correlation matrix means is to take the time series of every stock and scramble it in time completely. Now, if you make correlations where you randomly scramble every time series of every stock many times, all that you'll see in the correlation matrix are the random correlations that are possible just because of finite sampling and removing all the actual correlations that are there. So what this solid line here, marked here, shows is all the eigenvalues that are possible just by randomizing the correlation matrix. Now what it tells you is, in fact, the bulk of eigenvalues in the stock market matrix are explainable by just the problem of limited time sampling, and yet there are a few eigenvalues that stick out in the tail, and those are the ones that can be analyzed to be significant. And the interesting thing in this work is that it turns out that one can basically decompose the economy into the business sectors by looking at what stocks specifically contribute to each of those eigenvalues. So for example, the oil and gas sector is here, the automotive sector is here, and so on and so forth. So this is an approach mathematically to using correlation matrix to decompose the economy into business sectors. So the analogy to proteins is straightforward. Here we don't have a matrix of stocks by stocks and their performance in the market. Instead, we have a matrix about amino acids correlated to other amino acid positions in a protein, seen over the long-term divergence in evolution as sampled by the alignment of proteins we use in our statistical model. So again, in analogy to the work in the stock market, here's the eigenvalue decomposition of this matrix in black, and the red line represents the random estimates of eigenvalues that are possible by just scrambling every column of an alignment independently. And if you scramble every column of alignment independently, the analogy is the same as with the stock market. You will retain only the correlations that are possible just because of finite sampling, and you will remove all actual correlations between positions. And what this shows you is, again, the bulk of eigenvalues in the amino acid coevolution matrix, as we expect, are explainable by just the finite sampling problem of sequences, and there are a few eigenvalues that stick out in the tail beyond the noise, and we can analyze the pattern of residue correlations in those significant eigenvalues to deduce what is the interesting non-random information in this matrix about amino acid coevolution. And that produces a very interesting result. So by these approaches, these mathematical approaches, we can show that all of the statistically non-random information in this matrix is actually compressible into just 20% of the amino acid residues in this protein. So this is a roughly 100 amino acid protein. This is the PDZ domain. And it turns out that all of the, again, non-random amino acid coevolution signals seem to be held within a subset of amino acids, 20 of them or so. And within that 20, we see a hierarchical pattern of coevolution between the amino acids. And the direct implication of this is that 100 minus 20 or 80 of the other amino acids are, at least with the statistics that we have, look like they're evolving as if they're independent agents. They're not caring about the outcome of each other. And that's a not trivial and interesting result given the jigsaw puzzle-like nature of a protein structure. Here what it suggests is that many of those local environments are hardly noticing each other, but some group of amino acids in the protein really does care about each other, and they're experiencing a correlated pattern of evolution. So we gave this unit of coevolution a name. We call it a protein sector, and of course, you'll understand where that term comes from. It's a direct analogy to the idea of breaking down the economy into business sectors. Here we break down a protein into protein sectors. And the idea of a protein sector then is a unit of a protein family that is non-randomly co-evolving. The interesting thing is, though this unit was identified purely by statistical measures on sequences, there's a physical interpretation or a physical empirical finding which is quite interesting. And that is that if you put these 20% of the amino acids, the 20 amino acids in the PDZ domain, onto the three-dimensional structure of a member of this family, you find that it forms a physically connected network of atoms that links, in this case, the binding site through the protein core to a couple of surface exposed positions. So just to explain what I'm showing you here, I'm showing you two rotations of the PDZ domain. The residues that are in blue spheres constitute the 20% that are in this sector. And in the bottom two panels is precisely the same thing that's here, except now that I've also drawn in white spheres all the other amino acids that don't belong to the 20% that are here. And you see what I mean. The sector in this case describes residues that are directly at the binding site, and they disappear into the core of the protein and emerge again on a couple of surface sites that are at the backside of the protein. So this pattern of physical connectivity was not imposed by this method. And it's interesting because, again, we expect that all of the physics of interaction between atoms to occur through short-range forces, and we might expect that the pattern of coevolution between positions, if it was based in some physical property of the protein, might have a property that the positions would be linked through physical networks of interactions. The interesting thing, of course, is to ask is what do these sectors do in proteins? And I'm going to just show you, we're going to largely address the question of experimental testing of these models in the third lecture, but I'll show you a couple of examples just as a preview of what it means from a functional point of view. So in the case of the PDZ domain, it turns out that there is an example of a particular complex between a particular PDZ domain called the par6 PDZ domain and a regulatory molecule that acts on it called CDC42. The interesting thing is that CDC42 controls the binding of ligands to the active site of the par6 PDZ domain, but when the structure of the complex was done, it was found that CDC42 contacts this PDZ domain nowhere near the binding pocket, which is here, but contacts it on a backside surface. The interesting thing is the place where CDC42 interacts with the par6 PDZ domain is precisely the place at the back end of the protein where the sector analysis indicated were amino acids that were correlated in evolution with the binding pocket. And in fact, one can test these kind of these ideas further by making mutations at positions in the middle of this network and show that such mutations still retain the ability of this protein to bind the PDZ domain and still retain the ability of ligands to bind the active site, but the two are now uncoupled. One does not control the other. So at least anecdotally, there is some evidence that these systems of amino acids may be the underlying structure for what's called allosteric communication, or the ability of distant sites to control each other in proteins. So in the PDZ domain family, as you can see by this analysis, there appears to be just one sector. All of the non-random correlations are held within one single group of co-evolving positions, but that need not be true. It turns out that in some other proteins, such as the S1A serine proteases that I introduced in the first lecture, there's a possibility of there being multiple sort of quasi-independent sectors in a single protein. This is a bigger protein, having on the order of 260 residues or so, so the matrix is somewhat larger. But the interesting thing is the same methods that we described to decompose and analyze the information content of the matrix in this case indicates a decomposition where now there are three independently evolving portions of the protein. So as you can see, there are three groups of amino acid residues. Within each group, there's some hierarchical pattern of coevolution, but between the groups, it's as if the groups are almost independent of each other, as if there are three, as I say, independently evolving parts of this single protein. The interesting thing, of course, is that each of these groups, and we can just give them color names for right now. This is the blue sector, the green sector, and the red sector. Each one of these sectors obeys the property of physical connectivity in the protein structure. So each of these groups, red, green, and blue, comprise a physically connected network of atoms that are distributed throughout the protein structure. And what's interesting is that these three groups turn out to do distinct biochemical jobs in the protein family. The green group was already quite well understood. This is the basic catalytic machinery of the serine protease, the famous catalytic triad that mediates the actual hydrolysis of peptide bonds. The red group was a network of atoms that were built around the primary specificity site for substrates in the serine protease. And the blue group was, at the time the study was done, not known what this group does. But in experiments that were conducted to test this model of independence, and the nature of the experiment is to make a number of single mutations in the red sector and make a number of single mutations in the blue sector, and analyze each of those mutations for two properties: the effect on the catalytic power of the enzyme on the x-axis, and the effect of the mutations on the stability of the protein on the y-axis. And you can see the result. The closed circles represent mutations in the red sector, and the open circles represent the mutations in the blue sector, and the green dot represents the wild-type enzyme. And so the result was quite clear. It looked like the red sector affects catalytic power, but does very little to affect the stability of the enzyme, whereas the blue sector strongly affects stability, but hardly touches the catalytic power of the enzyme. So these biochemical data argued that this concept of independent sectors was consistent with biochemical independence of the properties mediated by those distinct sectors. More generally, one can carry out this sector analysis for a number of different proteins, and all I'll point out in this slide is that in classic model systems for signal transmission, like the receptors, and the classic problem of hemoglobin, allostery in hemoglobin, it appears that one can identify networks of co-evolving amino acid residues that are consistent with the known literature about the mechanism of action of these proteins. These are interesting results to begin with, and so let me just summarize what I've told you so far. What I described in this lecture is a statistical approach, a different approach to studying proteins, not by looking at the physics of individual proteins, by looking at the statistics of ensembles of sequences. We deduce a design for natural proteins in which the main units are these sparse but physically connected and collectively evolving groups of amino acids that we are calling sectors. And the sectors are embedded in an environment or otherwise weakly correlated amino acids. That's the pattern that we empirically observe by looking on the protein structure. And so the question that of course, all this begs is this really a good model for natural proteins? And what does it mean to say that it's a good model for natural proteins? As I pointed out at the beginning of these lectures, natural proteins display these following properties. They display the ability to fold up into a precise and ordered three-dimensional structure. They display the capacity to carry out biochemical activities. And they display the capacity to be adaptive, to be able to accept mutations that produce new functions. And then if we want to say that this model is a good model for natural proteins, we must show by experiment that this model can explain all of these properties of proteins. And what I would like to do in the next lecture is describe to you at least the first order set of experiments that have been done to try to test all of these properties with regard to this model of proteins that comes from the statistics of sequences.